Hello, my name is Jana Petermann and I'm presenting sub-project 3 that I'm planning to conduct together with Andreas Schult and with our Chinese partners Douglas Chesters and Mingqiang Wang. You will meet Andreas later. He will present sub-project Z2, and of course we will all be there for our discussion session. In this sub-project we're planning to look at plant herbivore predator food webs, stoichiometry and functions. So what do we know about plant herbivore predator food webs? We definitely know that plant diversity affects herbivores, for example, in terms of their species richness, their abundance, and composition. Many studies have shown this. On the left, you see just two examples from the China experiment, but many studies from different places have shown that plant diversity may affect herbivores. The effect strength and direction often varies, and this depends on, for example, whether specialist or generalist herbivores are studied. Of course, this also depends on plant traits, and this may also depend on time. So over time in an establishing forest ecosystem, these effects may also change. As soon as we move up the food web, I would say we know less on the effects of plant diversity on higher trophic levels. A few studies have shown effects, but some of these studies have also shown that the effects are weaker at higher trophic levels. And as soon as we leave the commonly recorded variables like abundance and species richness and look at things like biomass, we realize that there's very little data. Stocks may, of course, change as a response to plant diversity. However, we only know this really for the plant level, so plant diversity affects plant biomass, but we know a lot less how biomass stocks may change across the network. And of course, these stocks may change differentially, and this would then lead to shifting biomass pyramids with changes in plant diversity. Not only biomass stocks may change, also biomass flows or fluxes may change. We definitely know that trophic rates respond to plant diversity, and there are a few examples from the China experiment, but also from other places showing that, for example, herbivory or predation rates change as response to plant diversity. But how this translates into fluxes of biomass across trophic levels, we don't know. Other things that flow in food webs, for example, carbon or nutrients, we definitely know almost nothing on the effect of plant diversity on this multi-trophic stoichiometry. So far I've treated trophic levels as one entity. Of course, this isn't true. Trophic levels consist of several species that individually interact with one another. And these species-level food webs have also been shown to respond to plant diversity, for example, in structural properties like connectance or complementarity versus redundancy. But as you know, these species-level food webs are relatively difficult to collect, and so there are a few examples of tritrophic food webs in the literature. What are generally the research gaps that we identified for sub-project 3? As in other topics, there has been a focus on specific groups, collection methods, and vegetation strata. And the latter is very important, of course, if you think about a three-dimensional ecosystem such as a forest. As I said, we know less about tritrophic networks than we know about bitrophic networks, so we know less about higher trophic levels. And we definitely know very little about traits, biomass stocks and fluxes, and carbon and nutrients ratios, and how these change across trophic levels in response to plant diversity. But we will need all of this information to construct holistic energy and nutrient networks and look at how they change with plant diversity. And we're really only just starting to do this. This is a study from last year from the ENA experiment. So what are we planning to do in SP3? We're aiming to link food webs with biomass distributions and stoichiometry to truly predict responses of multi-trophic plant herbivore predator communities to tree diversity loss. And we split up this task into 3 work packages. Work package 1 is going to look at trophic community composition, specifically richness, abundance, body size, biomass, elemental contents. Work package 2 is going to look at trophic functions. Here we're going to measure herbivory and pathogen rates and also predation rates. And work package 3 is going to look at resolved food webs, and we'll will directly measure feeding length. So now I'm going to take you through those 3 work packages and explain briefly our hypotheses and our methods. Work package 1 on trophic community composition. Here we're hypothesizing that tree diversity effects decrease with increasing trophic level. And you can see this in our hypothetical graphs here with tree species richness on the x-axis and species richness, abundance, and individual size of our 3 trophic levels on the y-axis. In the first 2 graphs, you can see that trees are fixed in their species richness and abundance, but all other trophic levels are free to vary, and we're hypothesizing that they do vary, but that there are weaker effects the higher you go up the food web, and you see the shallower slopes here for predators. The methods we're using here are pitfall and flight interception traps on all core plots, and we're going to identify the organisms and also conduct body size measurements to get their biomass. In this work package, we're also hypothesizing that interspecific carbon-nutrient ratios of arthropods change with increasing tree diversity, and this is because of increased nutrient availability. And you can see the 3-dimensional graph here consisting of carbon content, nitrogen content, and phosphorus content, and each dot here is a species on a plot, and you can see that there are changes in elemental composition with tree species richness. We're going to measure these elemental contents for selected species in the core plots in work package 1, where we're focusing on 2 elements, carbon and nitrogen, and we're going to use many more elements in the VIP plots in work package 3. Together, we're thinking that these changes in abundance, species richness, individual size, and also elemental contents will then lead to differential changes in biomass across trophic levels, and thus to changes in biomass pyramids towards more bottom-heavy pyramids in higher tree species richness. Work package 2 will then deal with trophic functions, and here we're hypothesizing that energy networks show higher stocks and fluxes at higher tree diversity. And you can see this here, so with increasing tree richness, biomass stocks, but also fluxes will increase. We're going to use body size measurements from work package 1 here, and we're also measuring herbivory and pathogen rates using visual scans, as you can see here on the right, and predation rates using artificial caterpillars. Work package 3 will then look at resolved food webs, and here we're hypothesizing that structural properties such as connectins explain plant diversity effects on multitrophic functioning. You can see our hypothetical food webs in the center here and how they change with tree richness, for example, in terms of higher connectins. To collect these, we're going to use fogging or other tree-specific collection of arthropods on selected tree species in the VIP plots. We're also going to use additional information such as body size or literature data to get these higher trophic links here. But more importantly, we're going to use gut content barcoding of selected predators, and this will be done by our Chinese collaborators. We're well integrated into the overall project, so SP3 will provide arthropod diversity data to a number of sub-projects to complement their own collections. We're also going to work closely with SP2 to link soil, plant, and arthropod elemental contents, and SP3 will also receive additional seedling herbivory data and predation data of ants. Our Chinese partner project is very important. They're going to conduct the metabarcoding of spider gut contents. They're also currently building up a database of lepidopteran larvae and other organisms that will be very helpful for identification purposes. And they're also going to link arthropod occurrence data and prey of wasps for SP4. They have existing grants that help pay for these projects, but they have also new proposals planned for early next